Chapter 11 is all about gene regulation, and there's several different kinds of gene regulation. One of them is called transcriptional gene regulation, and this is handled differently in prokaryotes and eukaryotes. In prokaryotes, you often have genes that are related and are involved in the same process. For example, the lac genes are genes that are involved in utilizing lactose, the sugar lactose, as a carbon source. And this model, the operon model, is the model for how these related genes are transcribed. Often you have related genes co-transcribed, that is, the messenger RNA that encodes them is one long messenger RNA that includes all of the genes together in one messenger RNA. Instead of having one separate mRNA for each gene, you have one mRNA for several genes. And the way this works is that when there's lactose present, it binds to a repressor protein and the repressor falls off. So before you get to the part that encodes these proteins, you have two regions. One is called the promoter and the other is called the operator on the DNA. And it's part of the DNA sequence. And the RNA polymerase, the enzyme that synthesizes the mRNA, binds to the promoter. And you also have a second protein called the lac repressor. The lac repressor binds to the site on the DNA called the operator. The operator is just to the 3' side of the promoter, and it's before the start codon of the lac genes. So the repressor, normally the repressor is bound here, and it prevents the RNA polymerase from coming through, so it is blocking the RNA polymerase from transcribing these genes. But if lactose is present, it binds to the repressor, and that causes the repressor to fall off of the operator. And in that case, the lac genes can be transcribed, because now the repressor is not blocking the way, and the RNA polymerase comes through and it transcribes the genes. So this is an example of what's called a inducible operon, because it can be induced by the lactose. So normally this operon is turned off. But if lactose is present, it binds to the repressor and the repressor falls off. And that induces this operon and the genes get transcribed. That way, the genes are only transcribed when they're needed. Only when lactose is present do you need these gene products to break down the lactose and to bring it into the cell. Other operons are repressible operons, meaning that they're normally on all the time but can be turned off. And the textbook mentions one, the trip operon, which is normally on but can be turned off. And it's turned off by tryptophan being present. So in that case, the genes involved in tryptophan synthesis are normally on all the time, but if there's enough tryptophan being made by those genes, it feeds back and causes the the operon to shut down. In eukaryotes, the situation is different because in eukaryotes, normally only one gene is transcribed at a time, instead of many genes transcribed as a single mRNA. You have just one mRNA per gene. And normally, in the case of eukaryotes, the genes are activated by the binding of activator proteins to DNA sequences called enhancers. The enhancers aid in the binding of the RNA polymerase at the promoter. So the way that this works is that this segment will bend over, and so there'll be a bend in the DNA at this point. They'll bend over, and then they'll be able, once these bend over, they're able to interact at the promoter with RNA polymerase, and they enhance its ability to bind to the promoter. There are also a number of other levels of gene regulation besides transcriptional. There's chromatin structure, for one. That's the condensation of chromatin. So chromatin is the DNA of the chromosome being wound around proteins called histones. So these histones create a kind of structure that is kind of like a tetramer or octamer, and then the DNA winds around it several times. And that, if you have enough of those histones, the DNA can be tightly wound so that it can't be transcribed. So DNA that's condensed in that way is usually not transcriptionally active. So the degree to which the DNA is condensed is another way to control gene expression. And another way is called the alternative splicing of the mRNA. So the mRNA of eukaryotes consists of regions that encode protein, and in between those are regions that do not encode protein, and those are called introns. The exons can be combined in different ways. So imagine you have 3 exons and they're separated by introns. There's enzymes that will splice out the introns so that the exons can be combined. And the exons can be combined in different combinations. So for example, you can build one protein from a gene by combining exon 1 and 2, but you could also combine exon 1 and exon 3 and leave out exon 2, and that would be a different protein. Or you could have a third protein that it consists of exon 2 and exon 3 only, and not exon 1. So in eukaryotes you can get multiple proteins from the same gene. And this is why, although humans have only 20,000 genes, they have about 150,000 proteins, and that's because of this alternative splicing. It allows more than one protein to be made from the same gene. And this can be another level of control. Another level of control is at the translational level. So the translation of the protein can be affected by the proteins that interact with the ribosome, and that's another level of way to control. the gene expression. And the last one is the polypeptide activation and breakdown. So the polypeptide itself, once it's translated off the mRNA, often needs to be activated through some kind of proteolysis or some kind of modification of that polypeptide. In some cases, more than one polypeptide has to be combined, and so that you can control gene expression at that level as well, and also at the level of breakdown, so proteins can be broken down by proteases that can be also controlled. Chapter 12 is all about gene technology, and there's several kinds of gene technology. The first one we're going to talk about is the recombinant DNA technology, and this is the basic idea is to take a gene from one organism and place it in a different organism and express it in that other organism. And in the simplest example of this, you're taking a gene from humans, for example, the insulin gene, and placing it in bacteria and then expressing that gene in bacteria to produce large amounts of insulin, because bacteria can be grown in large amounts in bioreactors. So how does this work? So here we have this line represents the human genome, or one of the chromosomes, and on that chromosome is the insulin gene. And typically genes are surrounded by sequences that can be cut in a precise way. These are called restriction enzyme sites. And a gene will often have these sites outside of it on either side. And you can isolate the gene by cutting the DNA outside of the gene, provided that there's no site inside the gene. And so by carefully choosing these sites, you can find appropriate sites that will cut outside the gene but not inside the gene. And in that way you can isolate the whole gene. And then these restriction sites, the way they work is they are typically 6 nucleotides long, and they have what's called a dyad symmetry, which means that they read the same on both strands but in opposite directions. So here, for example, we have one called EcoR1, and EcoR1 has the sequence, the site has the sequence GAATTC, and that's in the 5' to 3' direction. On the opposite strand, you have the same sequence GAATTC, but in the opposite direction. So 5' to 3' again, but on the complementary strand. So these sites have dyad symmetry. They read the same backwards and forwards. And so certain enzymes can recognize that dyad symmetry and then cut at the last nucleotide on both sides of that. And these enzymes, these restriction enzymes, are used by bacteria as a kind of defense against viruses and other bacteria. And but we can utilize these enzymes to manipulate DNA sequences and to take genes and place them in other organisms where we can mass produce the genes or their products. So the way that works is the restriction enzyme sites, when they're cut, the covalent bond between the phosphodiester linkage between the nucleotides is cut, but it's only cut at one nucleotide on each strand, and when that happens, it leaves an overhang. So here, when it cuts this strand, you get this. So you get the G on the upper strand, and on the lower strand you have CTTAA. And on the other strand, you're left with CTTAA on the upper strand and a G on the lower strand. So it's basically cutting here, and then this is all intact, and then there's a second cut here. So that leaves this portion shown here, and this portion shown over here. And then these lines represent the remaining part on either side. So bacteria have small circular DNA molecules called plasmids, and these are autonomously replicating, so they replicate independently of the chromosome, and they have these restriction sites in them, or can be modified to have these sites. And so imagine you have a plasmid with a single EcoR1 site, and you cut it with the EcoR1 enzyme, then you're going to open up this plasmid as shown here. So you have this overhang here and the other overhang on the other side. But notice that if we cut out the gene out of the human chromosome with that same enzyme, we have the same sites on the ends. And you'll notice that these overhangs are complementary to the overhangs on the lower strand. So this overhang here, AATT, will hydrogen bond with the overhang on the lower strand, TTAA. And on the other side of the gene, likewise, you have the overhang TTAA, and that will hydrogen bond to the AATT overhang. And so that in this way, you can move this gene into the plasmid, and it will hydrogen bond to these overhangs in the plasmid. And then so that stabilizes it, and then another enzyme called ligase seals the gap, seals that diester bond to form the covalent bonds needed to make that completely stable and place it in there permanently. It's not quite as simple as that, because you do have to remove the introns from a human gene or any other eukaryotic gene, but there's a way to do that. You can remove the introns by, instead of starting with the DNA, you can start with the mRNA, and there's an enzyme that can make a copy of the mRNA as DNA called reverse transcriptase, and that enzyme can be used to get a copy of the gene that lacks the introns. So it's just the exons, and you put that in, and then you use the ligase to seal it, and then that modified plasmid can then be put back into the bacteria and used to express the insulin. So this is an example, just one example of recombinant DNA technology, but it's can be used for many things besides insulin. It's often these days used for many commercial enzymes to produce large amounts of it. Another important concept is the concept of DNA hybridization, or this is sometimes called DNA probes or DNA primers. So if you wanted to find a particular sequence of DNA, say you're interested in a particular gene or a region that's being used for forensics, you can find it using these DNA probes or primers. What they are small segments of DNA that are synthesized chemically, and you can synthesize any sequence you want, and it will bind to the DNA of the sample, whether it's DNA from an organism that you're studying or a crime scene sample, something like that. You can design a sequence. Usually they're a little longer than this. They're usually about 20 base pairs long, but they will specifically bind, hydrogen bond to the sequence they're complementary to. So if this were a sequence you were interested in, you could design a sequence that's complementary to this one and then use it. So for example, if this were complementary to the sequence you want to amplify, you could design it so that, let's say you were interested in this and whatever is to the left of that, you would design it this way, reading 5' to 3', and then this available 3' end becomes the place at which the DNA polymerase will start to synthesize the strand. And you can do the same thing on the opposite of strand. So you can have a sequence that is further down from there, and you can amplify that sequence as well. So you can get the product of both of those, and wherever they overlap, that's the part that will get amplified. And I'll go into the details of that in a minute. That's called a PCR method. But this method is used to kind of home in on whatever you're interested in. And so that concept of DNA hybridization is very important. And it's the whole basis for the polymerase chain reaction, which I'll get to in a minute. Also important is the concept of agarose gel electrophoresis. So let's say you have a DNA sample that's a mixture of fragments, and you want to know which fragments are in this mixture. You can separate them through a gel which is made of agarose, and if you apply an electric current through this gel, it will separate the DNA. So you load the DNA in a little well in this gel, and then you apply an electric field and the electric current through the gel, and the DNA will move away from the negative electrode and towards the positive electrode, because the DNA, as we know, is negatively charged from all the phosphate groups. But the fragments will separate by size, so the smallest fragments will move faster through the gel than the medium size or large fragments, and in this way you can separate them out by size. Another technique used very often in gene technology is called the polymerase chain reaction, or PCR for short. And this method is used to amplify small amounts of DNA of a very specific sequence. So let's say that you want to study a particular gene, but you only have a limited amount of DNA. You can amplify just that one gene from your sample. Another thing you can do is to amplify certain repetitive sequences from crime scene samples. So certain repetitive sequences can be used to identify individuals, and if you have a very small amount of material from a crime scene, like a single hair or a single stain on clothing, you can amplify that with this method and get large amounts of the DNA that you can then analyze further. So this illustrates how this works. So here's a gene of interest. What you do is you design primers, those short segments that I mentioned earlier, and you design them to be complementary to the DNA line just outside the gene on either strand. So this one is complementary to the top strand, this one is complementary to the bottom strand. And then what you do is you just take the DNA and you heat it, and that the heat will denature the strands. It'll separate the two strands, and once they're separated, you can add the primers, and they will bind wherever they're complementary to. And by adjusting the temperature, you can get that to be very specific. And at the specific temperature that they bind, that they hybridize, they'll bind just to that sequence and no other sequence. Then the next step is to add the DNA polymerase, and the DNA polymerase recognizes the end of the primer, the 3' hydroxyl group at the end, and synthesizes the complementary strand by the complementary base pair sequencing starting at the 3' hydroxyl. So that occurs on both strands that were separated here. So now what you have is you've gone from a single double-stranded DNA molecule to two complete double-stranded DNA molecules. One molecule to two molecules. And then to get more of it, you just repeat this process again. So in the next cycle, you'll go from 2 to 4. And if you repeat it again, you'll go from 4 to 8, and then 8 to 16, and 16 to 32, etc. If you do just about 40 of these cycles, you'll have millions of copies, because if in each cycle the number of copies doubles. So there's an exponential increase in the number of copies, and by about 40 cycles you have millions of copies of that very specific sequence where the primer is bound. And this can be used to study genes. It can also be used, as I said, to analyze the crime scene samples. And finally, some proteins of medicinal value that have been mass-produced through recombinant DNA technology. So here I'm just going to list a few of these. So there are others as well, but here just to list a few is insulin. So insulin is the protein being made, and its function is to control blood sugar, and it's mass-produced in bacteria, as I described earlier. Another one is growth hormone. So if someone has a defect in growth, then this can be provided and controls body growth, and it also is produced in bacteria. Third one is hepatitis B vaccine, and this is used to vaccinate against a particular virus, the hepatitis B virus, and this one is produced in yeast. So these are just a few examples. Others would be the clotting proteins that I talked about earlier when I talked about hemophilia. This one of the clotting factors is missing in hemophiliacs, and that can also be provided through this technology.